to this clinician scientist series actually. Though it started as a very small group, we see the numbers increasing, and we see some people who are not initially interested on this basic science research solely. They seem to be some trickle, and I attribute this entirely to Dr. Narasimhan's effort, who has got a tremendous understanding of basic science in addition to the clinical knowledge. So as a part of this, we are trying to incorporate sessions which have got clinical things and also basic science, just to make sure that we create some interest among the other clinicians to make sure that they come into clinical practice. So this is we're going live on this actually. There is a huge viewership for this. We're having some industry people also watching us this. So this time we have a very interesting thing as what the cardiologists have been doing behind the scenes. We never realized it till they showed it to me, that saying that this is what is being done. It is a tremendous thing to see what instrumentation is being done and how effectively it is being used. Kudos to the entire cardiology team and the cardiothoracic team which have started this entire concept. And within a short, I don't call it short, but it's in a relatively short period of time, they're able to translate it into the clinical side. This is the biggest advantage that we think that we have it in AIG compared to any other place. So the fascinating story, I think we'll hear it from the concerned people. This is really fascinating actually. You should see to watch what is being done. Sukesh, Dr. Narasimhan, the podium is all yours.
Good afternoon, everyone. It's very important. I don't know how many of you, some of you are not in this heart failure space. When the heart muscle becomes diseased, either because of blood flow obstruction or disease due to infection, myocarditis, the only thing that was there for the last decade were few drugs like digoxin, which started several years ago. And then for long, the treatment of heart failure was not anything to do with the heart. What they tried to do was reduce the workload on the heart, take the preload by giving diuretics, or rest the heart by giving a drug like beta blocker, or unload the heart by blocking all the counter-regulatory hormones. But that itself translated into humongous amount of mortality benefit and morbidity. So with the result, you have increasing number of patients surviving severe forms of heart disease and who are desperate to have some destination therapy. I think I should congratulate both Dr. Reddy and Dr. G.V. Rao to conceive and make this fertile ground for both research and translational work. And that kind of an atmosphere is a big advantage for youngsters getting into the field. And several centers in Europe, like Max Planck Institute, some of the centers in the US, the physician scientists are the ones who will lead the way. And to this end, I should really congratulate both Dr. Sukesh and Dr. Roshni, as well as Naresh, for taking the initiative and working on indigenous products as a form of mechanical circulatory support. It's a long struggle to do it for the first time, and bear with me, they've come a long way, and it's soon to enter the clinical space. But before we proceed with the presentation by Dr. Sukesh and Roshni, I would request my colleague Dr. Auris to kind of give a bird's eye view. What are we talking about? What is the magnitude of the problem? It's humongous in the country, like 1 billion population. We are going to see more and more of patients surviving heart attacks, surviving other forms of heart disease, ending up with ultimately with severe heart failure. Auris, can you probably?
For the introduction, we'll get started with All right, I'll keep this as a brief introduction. We'll just talk about the magnitude of the heart failure problem that we have. If you look at the epidemiology of heart failure, a lot of this data is from Western countries. We have some data from India, but apart from the obvious health morbidities and mortalities, this is one of the most important problems facing medicine today, because as we have gone through decades of increased survival from heart attacks, heart attack care has really improved, starting with thrombolysis and stenting and so forth. That now there are so many people living with heart failure that the cost implications are enormous. Now, you can imagine this was a decade ago. Data from a decade ago is $30 billion estimate for heart failure, and then this is going to projected to increase to $70 billion. This is sort of the global landscape. And you will see that in countries like India, you may not even be able to spend this kind of money. So the magnitude of problem is even larger if you look at it. Because when you look at economic costs, you're only registering what has been spent. This is a good slide to summarize, that there are about 6 million people in the US with heart failure. Globally, it's estimated to be about 20 to 30 million. And we don't think of this. We write heart failure very frequently, but a heart failure diagnosis is actually kind of like a cancer diagnosis. Advanced heart failure. You should be thinking of it like that, because if you look at the median survival, it's still less than 2 or 3 years in the absence of advanced therapies. And heart failure is a very broad term. I think the focus of today's meet is more advanced heart failure, where there's about 10% of patients where you're trying to use advanced therapies that include mechanical circulatory support and organ transplantation, which we are not going to talk about today. And the mortality globally is probably upwards of 1 million. This data of 350,000 is just from the US alone. And this is one summary slide. Again, like I said, the data from India are scarce, but this was a paper published in 2011. And the prevalence in India is about 1.8% or so. Now you can imagine, convert this or multiply this with our population, and we are not even skimming the surface of the problem currently. Again, another slide to show you that the median survival for heart failure is not good. So this is first hospitalization. People have a median survival of 2.5 years. And by the time these patients come and get admitted 3 or 4 times, their survival rate is expected to be less than a year, half a year or so. That's why in the heart failure world, about 2 hospitalizations is considered an ominous sign for patients with advanced heart failure. And that's generally the time that referral for advanced therapies should be considered. And the other context to look at heart failure is more recently trying to classify this, the American College of Cardiology and the American Heart Association classification is stage A to stage D. In the clinical paradigm in cardiology, now stage A means at-risk patients. So these are all patients who could potentially become heart failure patients tomorrow. So you have to gear up your basic and clinical translational work to meet this demand 20 years down the line. And these are patients at risk for heart failure, but they already have heart disease. They've had a heart attack, there's scarred tissue, the pump is not good, but they're doing okay. That's stage B. Stage A is people with just high blood pressure, diabetes, who are all at risk of developing heart failure. Stage B is they're doing okay with minimal therapies. But stage C and stage D is where the clinical teams get involved. Stage C is where majority of the cardiologists are involved, and stage D is multidisciplinary, and the surgeons come into the picture with advanced therapies of LVADs and heart transplants. So again, just one slide to emphasize that stage A and stage B, which are more of the, this is what the future holds. And for this, it's estimated that there's 120 million people with hypertension now, and close to 200 in a couple of years in India alone. 65 million people with diabetes, 60 million people with coronary disease. So even if 10% of this trickles down into advanced heart failure over the next decade, 2 decades, there's a lot of people waiting to be served. This is a very famous slide published from the New England Journal of Medicine, 2003. Marion Jessup, one of the leaders in heart failure transplant cardiology from University of Pennsylvania. So if you look at these stages, it translates clinically like this. So at stage A, you're just trying to control the risk factors, educate patients, let them know what is in store if they don't correct these modifiable risk factors. Stage B, they already have problems and you need to start medical therapy. And this includes ACE inhibitors or angiotensin receptor blockers. Stage C is where we now have a host of medical therapies. I won't go into the clinical details of it. And then stage D is the advanced sort of phase where they need medications, frequent ICU hospitalizations, medications to increase their blood pressure, increase the pumping. As Dr. Narasimhan said, this is the stage where you're trying to work on the pump itself. You've already optimized the preload to the heart by reducing, fluid in the body by diuretics, reducing the afterload, that is the pressure the heart has to beat against, blood pressure and the kidneys. That's with the angiotensin receptor blockers and the newer drugs called ARNIs, as we call it. But our focus again today is more on the VAD section. This is a slide just to let you know of all the advances in pharmacology that have happened over the past several decades. But the past decade alone has been very instrumental. Highlighted in blue are all these medications that have been available to us as clinicians the past decade alone. And then you'll see that the first heart failure therapy, the ACE inhibitor, was 95, and there was relative stagnation in the early 2000s. But this is the result of a lot of basic science and translational work in the immunology and heart failure animal model space. And so this is sort of a standard slide where current advanced heart failure reduced. These are patients where the pump is weak. We have a host of medications with known mortality benefit, and we have a host of medications where we can supplement based on a specific clinical profile of the patient. And if you look at any heart failure patient, they have initial hospitalizations, initial diagnosis, then they do pretty well with time. You see the functional status, they do pretty well. There's this false sense of well-being that the patient experiences. They think that the problem is resolved, they got admitted, it got resolved, but that's never true. It's going to happen again. Once you have a problem, it's a constant process. And you'll notice that with time, they have further deteriorations in functional status. So if you want to refer them and have them do well with an LVAD or transplant, it's in this sort of finding the timing right in this section where they're doing well, but you know that they'll go down based on all the information. Intervening then is going to give them a better outcome, as opposed to what typically happens now in India, where you see these end-stage people, they're dying in a hospital, and then you're doing emergency ventricular assist devices and transplants, and the outcomes here are not that good as if you do it at the right time. And then just to end this talk, again, so you may have some advanced heart failure that you're seeing in the clinic, but at any point they may deteriorate and get admitted with reduced function, what we call cardiogenic shock, where they can't maintain their own blood pressure. And then you have to assess them either in the acute phase or the chronic phase where they're relatively stable for these advanced therapies. And what are these advanced therapies that we talk about? Temporary mechanical circulatory support. These are pumps that are only temporary. The patient cannot go home with them. This is to keep the patient alive and support the blood flow in the hospital. And I'll just show you a couple of pictures of what we have done here. And then to be able to give them support devices that will allow them to function at home, those are what we call LVADs, or durable mechanical circulatory support. Dr. Roshni will talk about this a little bit. And then we have the traditional heart transplant, which has been around since the 1970s and currently in a very good phase where there's more than 90% survival. The issue is of supply. There's only globally about 6,000 hearts or so that are available for donation, although the people waiting for such advanced therapies exceeds about 20,000. So this is where there is a gap where devices, which there is no limit on manufacturing, if you are able to make it cost effective, you could help a lot of patients. So these are some of the cases that we have done here. This is just a simple balloon pump, which gives you about 1 liter per minute of support. So patients who present with shock, this is an initial temporizing measure, relatively inexpensive compared to the other therapies in this space. We have done a few of these devices. This is called an Impella. This is a pump that goes into the heart and takes blood out because the heart can't squeeze that blood out by itself. And then it puts it into the aorta, sort of artificially supporting the heart, the left ventricular pump, but this is again temporary. Patient is attached to a console like this. It can stay in for about 2 to 3 weeks at the most. Currently, cost prohibitive, costs more than a heart transplant. So just the cost of the device as well. And this is another therapy called an ECMO device, which is an extracorporeal membrane oxygenator. Can be done percutaneously or surgically. A little less expensive than the Impella, but again, is able to support the heart with up to 5 liters per minute. So basically supplants the entire cardiac function. And this is how critically ill these patients are. This is a patient that we put on ECMOs, kept him for 2 weeks, recovered. Again, he's in that phase where he is doing okay, but he's young. So at some point he will deteriorate. So at some point in the future, he may need a ventricular assist device or transplant. And this is, a picture of a left ventricular assist device that we did recently, in addition to, a couple of heart transplants that we have done. So this space is growing at AIG, and I think the fact that we are doing this meeting here is the indicative of the future interest in the cardiovascular world, but specifically here at AIG. I'll end with this talk. Thank you.
Thank you, Dr. Ravi, for giving a bird's eye view of heart failure and all the options which are available for treating such patients. First of all, I would like to thank Dr. Jivirav Garu, sir, for providing this platform where we have an opportunity to interact with, the clinicians who are able to meet the scientists. This will help research flow from the lab into the clinical practice. Okay, at AIG, we are also very actively involved in management of heart failure. And if you see, in the 1980s and 1990s, a lot of research went into cardiopulmonary bypass and then cardiopulmonary bypass surgeries, valves, and those kind of things. But today, the most happening thing, research is all about, mechanical circulatory support. As Arus have rightly pointed out, there are only 6,000 hearts available per year, and more than 20,000 patients are waiting for a heart transplant. And these patients, if they have to live, they have to go on some sort of an assist devices to continue their life. So at AIG, we are actually involved not only in treating advanced heart failure patients, but we are also involved in developing devices which are cost effective, and then it will be more helpful for Indian population. Now I request Roshni to present what all the mechanical circulatory support devices which are available with us today.
Good afternoon, everyone. So after the extensive discussion regarding the magnitude of heart failure problem, so we as cardiac surgeons are concentrating on the circulatory support devices, which will help the patient to continue their limited lifespan until a transplanted heart becomes available. And these circulatory devices have been a revolution in the treatment of end-stage heart failure. So how these mechanical circulatory devices help is they are either designed to assist or replace the function of left ventricle or right ventricle or both. So these devices are indicated to provide hemodynamic support to patients with critical cardiogenic shock or symptomatic advanced heart failure, which is refractory to guideline-directed medical care. It decreases the workload on the heart while maintaining adequate flow and blood pressure. So what are the indications of mechanical circulatory support devices? So we have characterized patients into various categories based on their cardiac status. So usually we consider the patients as bridge to decision. Bridge to decision is when a patient is having refractory cardiac shock where there's no scope for survival, but to bridge the patient so that we give some treatment option available for him. The next is bridge to candidacy. Some patients are sick so as they are not even eligible for cardiac transplant or any other thing. So in such situations, we bridge the patient with a mechanical circulatory support just to make him eligible for further transplant. And circulatory support devices are also used as a bridge to transplant, wherein the heart becomes available, and meanwhile we provide circulatory support. And in some cases where the patients have refractory heart failure secondary to reversible conditions, sometimes such as myocarditis, we use these devices as a bridge to recovery. And now the latest circulatory devices have become destination therapies where patients live with these circulatory devices inside them without having to wait for a transplant. So as Narasimhan sir had already said, the initial focus of heart failure patients was to unload the stress on the heart. If you think of horse as the heart, so initial treatment strategies was to unload the stress, slow down the heart. So where the MCS helps is either get a new horse or get a tractor. That means provide additional support to the heart. So how are these mechanical circulatory support devices categorized? So first thing is based on the location of the pumping chamber, and then based on the type of ventricle supported, either single ventricle or both, and their pumping mechanisms, and the indicated duration of support, as Dr. Ores said, sometimes it's temporary and sometimes it's long-term. So usually the pump location can be inside the body or outside the body. So the terminology for that is extracorporeal, where it is outside the body. Paracorporeal is outside but adjacent to the body. Intracorporeal is inside the body, and orthotopic is in the natural position of the heart. So these are the various kind of MCS devices which we are using in clinical practice. So few of them I have listed here. So intra-aortic balloon pumps, extracorporeal membrane oxygenators, ventricular assist devices, total artificial hearts, and Impella devices. So I'll be discussing each one of them. So in these, the short-term mechanical circulatory devices are the intra-aortic balloon pump, Impella devices, and pulsatile heart pumps, tandem hearts, and ECMO. I'll be discussing further details about it. There are a few long-term devices which are left ventricular assist devices and total artificial heart devices. So the first and foremost is intra-aortic balloon pump, which is used in almost many cardiac emergencies where patient has refractory cardiogenic shock. So this machine was invented by American cardiac surgeon Mr. Adrian Kantrowitz, and this machine has saved countless lives throughout the world because it is cheap, it's easily available, and it provides immediate cardiac support. So it works on a principle of counter pulsation. So a balloon pump, this catheter is inserted percutaneously through the femoral artery, and it is placed into the descending aorta. So what happens is, this pump deflates during systole, causing reduced afterload and increases the cardiac output, and inflates during diastole, causing increased coronary perfusion and increases the diastolic blood pressure. So as you can see, this machine works on a principle of helium gas. It provides a pneumatic counter pulsation, and the most commonly used device worldwide to provide temporary cardiac support. So next is extracorporeal membrane oxygenation. So this provides partial or complete support to both the circulatory and the respiratory systems. So blood is actively oxygenated and decarboxylated using a membrane lung which is outside the body. So it has an oxygenator which temporarily helps to function both as a lung and heart. So there are two types of extracorporeal membrane oxygenators. So when we need only lung support, we use a VV ECMO, veno-venous ECMO. And when cardiac support is also necessary, there is a veno-arterial ECMO which is in place. So how the veno-venous ECMO helps is, so we drain blood from venous system, usually from a femoral vein, and then oxygenate the blood and reinfuse it into another venous system, mostly internal jugular vein. So it gives rest to the lung while the lung is recovering. In the veno-arterial ECMO, we take blood from a peripheral vein such as femoral vein, oxygenate it, and then give it back to an artery. So it helps in the circulatory support also. So this was the veno-arterial ECMO which we had placed in a patient recently. So when there is a critical cardiogenic shock, so the veno-arterial ECMO gives support both for the lung and for the heart. So another modification of the ECMO is CentriMac. So all these previous ECMO devices, they were known to cause hemolysis because this is an extracorporeal chamber. The blood is circulating into the outside the body. So further advances have come in this, wherein a CentriMac pump has been developed, which works on a magnetic levitation technology, causing less hemolysis, and it can be used for a longer duration of time. And patients have been supported on these pumps for more than a month, and it can support both the left and right ventricle. So next, Impella, as Dr. Oris had already said, it's a catheter mounted microaxial pump that uses the Archimedes screw principle. So it pumps blood between an inlet and an outlet, and various modifications have been done in this device to provide even complete support to the heart. So as you can see, there is a catheter which is placed from the femoral artery into the left ventricle. So it takes blood from the left ventricle and delivers it into the ascending aorta, thereby reducing the stress on the heart. And even for the right ventricular support, it can be done in the similar way, wherein the catheter is placed between the IVC and the pulmonary artery. So blood is drawn from the IVC and delivered into the pulmonary artery directly. So next is tandem heart. It is similar to Impella devices, but it gives only the left heart support. It is used as a left heart bypass device. So it has an external pump, which is again magnetically levitated. It's a continuous flow pump, which can be used for both, usually used for left heart bypass. So as you can see, the catheter is inserted from the right atrium into the left atrium, and then it is delivered into an external artery, such as a femoral artery. So blood is withdrawn from the left atrium and delivered into a peripheral artery. So now the we have completed the temporary devices. Now the long-term durable devices. So first I would like to talk about left ventricular assist devices. So these are again continuous flow pumps, usually implanted intrapericardially, and they provide support to the left ventricle, and they provide a full duration of cardiac output support, and they can be managed for a long time. So the commonly used devices commercially available in the market are HeartMate 2, HeartWare HVAD, and HeartMate 3. The most recent technologically advanced pump is HeartMate 3. It is a centrifugal flow pump with rotates about a speed of 5,000 to 6,000 rotations per minute and can deliver about 4 to 6 liters of cardiac output. So this was the operative image of a patient in whom we had recently implanted an LVAD. He had a end-stage heart failure. His blood group was B negative. He was enlisted for heart transplant, but since the heart was not available, we considered this option. So as you can see, there is a pump connected to the left ventricle here. So it takes the blood from there, and through this tube, the blood is directly delivered into the ascending aorta. And there's a long-term mechanical circulatory support device which works, has worked even for 10 years or longer. And patients are able to carry out the normal activities with these pumps. So the last in this is total artificial heart. So in this, the pump mechanism is there is a pulsatile flow with a volume displacement. It is implanted in the intrapericardial orthotopic placement. So as you can see, both the ventricles are replaced by an artificial membrane, and with the pneumatic pulsation, the blood is pumped into the great arteries directly. So there are two types of total artificial heart commercially available in the market right now, but these are quite expensive. So in the Indian market, it has not yet picked up that much. So to conclude, the global mechanical circulatory support devices market size was valued at US $1.1 billion in 2021 and is poised to grow at a significant compound annual growth rate of 9.5% during the forecast period of 2022 to 28. And there's a need for research and development initiatives to advance present technologies in view of scarcity of cardiac donors for transplants, requiring the design of safe physiologically adaptive MCS systems. And there's a scope for further research to make the devices cost effective, especially to cater to Indian population, as all these devices are extremely expensive. So with this, I would like to conclude my presentation. Thank you.
Yeah, thank you, Roshni. Now I request Sadia to present about the Indus pump. So Indus pump is basically a ECMO, I mean, like basically a left ventricular assist devices which is externally placed, which has been developed in collaboration with Share India and with AIG and with other engineering institutes. We're still in the preliminary stage, and then I request Sadia to present about our Indus pump.
Good afternoon, everyone. I'm Sadia Alvi. I'm an assistant professor in Mechanical Engineering Department of Srinidhi Institute of Science and Technology. So today I'll be presenting you about the development of Indus, which is a indigenously developed ventricular assist device. So as my colleagues already discussed about how a ventricular assist device works, so I'll quickly jump on to how we have developed our product. So as it was discussed by Dr. Roshni also, that the cost of these devices have made a major cause of concern in India, because the cardiac patients are a lot, but devices, the cost of the device and the surgery is a lot more. So we have developed Indus, which is a ventricular assist device with the features of magnetically levitated impeller with a maximum flow rate of 10 liter per minute, maximum pressure of approximately 750 mmHg, and a maximum speed of 5500 rpm. This is very similar to the already available Centrimag in the market. So the cost of this device is proposed to be about 25 to 35% of all the imported LVADs. So when we develop any device completely indigenously instead of importing it, the cost drastically reduces. So the objectives of our project are to design and analyze the model using softwares, then rapid prototyping using 3D printing techniques, preliminary in vitro hydrodynamic and hemolysis test, manufacturing of the product after all these tests are done, then assembly of the product, in vitro biocompatibility testing of the injection molded pump, then in vivo testing on the sheep. After we are done with in vivo testing on sheep, only then can we go ahead with clinical trials and commercialization of the product. These are the various components of Indus pump. So the first component is the top casing, then we have a bottom casing, then the impeller, its cover, and a permanent magnet that sits inside the impeller. This is a very lightweight device. It would weigh roughly about 60 grams. As you can see, the magnet is placed inside the impeller, and the impeller sits in the well of the bottom casing. So the material used for the manufacturing of this device is medical grade polycarbonate. So first we design the components in the softwares. Then we do the CFD analysis. So designing is the easiest part. We can design anything and everything that we want to do in the softwares. But analysis has to be done for the fluid dynamics, where the engineering comes into picture. We cannot directly manufacture any component with the design that we get. The fluid dynamics, the pressures, the flow rates, and the shear stresses need to be evaluated so that the blood does not get damaged. So the second step is the CFD analysis. After we are done with the analysis, the parts are 3D printed in the laboratories. So this is a photopolymer resin which is used to 3D print. Then the testing is done on the 3D printed prototypes before we proceed for the manufacturing. And only after we get satisfactory results, we proceed for manufacturing. And this is the final injection molded pump. As you can see in the pictures, this is the injection molding machine which is used to draw out these pump parts. So proper care has to be taken so that there is no mold release involved in it that can cause any kind of blood damage. So Vasanta Tool Crafts is a leading manufacturer which is in collaboration with Share India and AIG Hospitals. They are helping us out for the manufacturing of this device. So in AIG, we have got a dedicated research lab for conducting in vitro bench testing on these injection molded parts. So on the right hand side, you can see a mock loop is set up in this fashion where the inlet of the pump is connected to a reservoir, and then there is a flow sensor and two pressure sensors which are connected to the loop. So any kind of test pump can be connected in this loop to conduct hydrodynamic hemolysis, activated thrombosis, and von Willebrand factor tests. So this is the test setup in AIG Hospital for the hydrodynamic testing. So hydrodynamic testing is basically used to check the pump performance. So here we make the HQ curves or the pressure versus flow rate curves for the pump. Only after we get satisfactory HQ curves, we'll proceed with the biocompatibility testing. In this image, you can see there's the heating water bath in which the reservoir is placed. So we are mimicking the human blood circulatory system here. Therefore, we are maintaining the temperature at 37 degrees centigrade. So these are some of the HQ curves. So we have conducted multiple HQ curves, but these are two of the latest HQ curves that we have got. So it is in comparison with the Centrimag pump. So Centrimag is already established commercial pump. So you can see here that the curves are in close approximation with the Centrimag, especially at low RPMs. Next, we come to hemolysis testing. For hemolysis testing, we use bovine blood or human blood to check how much blood is getting damaged with using this kind of mechanical pumps. So as it is a centrifugal pump, it undergoes a lot of shear, and there is a chance of blood to get damaged and lead to hemolysis. So we conduct these tests where we take out blood samples from the loop every 1 hour and check for the plasma-free hemoglobin that is present in the plasma. So, and in the end of the test, an index is calculated known as the normalized index of hemolysis, and for that value, a 0.01 is the acceptable limit. So, as you can see, for Indus, and even for the 3D printed prototype, the values that we got is way below the acceptable limit of 0.01. And for injection molded part, we even got better values of 0.0018, which is in close proximity with Centrimag. Other tests that we conduct are the von Willebrand factor and activated thrombosis tests, which are still ongoing in AIG Hospital. For the von Willebrand factor test, we use platelet poor plasma, and for the activated thrombosis, we use blood. Usually, bovine blood is used for these tests. So, VWF is a protein that is essential for the clotting mechanism in the body. So, again, using pumps like these can depreciate the quantity of VWF present in the body. So, tests need to be conducted before we proceed for any kind of animal or human trials. So, ELISA tests and Western blots are done in AIG, AHF Foundation also. And activated thrombosis test is used to test where at which locations the thrombus formation takes place in the pump. So for that we have also designed a particular clamp on device which can reduce ring thrombus in the device. So this was all about the Indus pump, but to drive the pump we also need a motor. So the main components of this entire device is a pump, a motor and a console. But currently we are using the Indus pump with the Centrimag motor and controller as it is fully compatible with it. So we are now also proposing to develop the motor which will be specifically for Indus and it will be totally indigenously developed. This will further reduce the cost of the entire system. So it is based on a magnetic levitation principle and a console is going to be attached to it, which is similar to the Centrimag console, which is currently ongoing. The progress is still happening in this console area. The motor driving mechanism has been developed. So we expect to develop the motor and the console by end of this year. So there are a lot of institutes that are involved in this project. Prestigious universities like Cornell University, University of Pittsburgh, and Integris and Share USA are involved. And from India, there are many medical institutes like AIG, Share India, Palamuru Biosciences, and other engineering institutes. We have a team of medical experts, industry experts, and academia who have collaborated with us for this project. Thank you all.
From what we use.
We get it from Palmur Lab, sir. You know,
Already
we send,
collected.
one day before, we send a person to Palmur Lab and he collects it in a bag. We provided the bags which we use here, and then they put heparin in it, and then they come and then CPD, sorry.
But it is similar to what we.
Absolutely, yeah. So, believe me, it is not easy. It is a Herculean task. For the last 7 years, I still remember 2017, when both my children got married, PS called us for a big meeting with all the people from US and other institutes coming and all the stuff. And for the last 7-8 years, we've been struggling. Every week on a Saturday, we have a Zoom meeting, we talk about it. And big people like even Jim Long is involved in the project of developing, and we have reached this stage. I think we have reached approximately 70% of it. So basically, the three components of the pump. One is the impeller. So this is the impeller. Basically, it's a magnetically levitated pump. Like basically centrifugal pump. It draws blood, and then it pumps it back. Okay, and then this can fit onto the motor of a Centrimag pump actually. And then actually the motor is also being developed, and I think it should be ready by this year end. And there's also a controller, which shows how much flows you're flying, what is the RMP and all the stuff, and pulse dial index and all the stuff which shows up. So it's going to take a while. So in the meantime, when this pump becomes available for us, we need to test it. So it has to go through a series of processes, like before it gets approved for clinical use. So first it has to go into animals, and then we have to make sure all the data is right, and then we have to go for clinical trial. So we searched so many facilities in and around Hyderabad, and finally we zeroed on Palmur Lab. Palmur Lab is a huge big animal lab, and we have sheep, we have pigs. Me and Naresh did a lot of experiments, even with pacemakers also there. And then Naresh is also very actively involved. He's the main person who puts all the pumps. I only guide him to do all those things. So in Palmur Lab, we conducted animal experiments, but not with this. We conduct with Centrimag, because we have to master the technique how to put the sheep or a pig on cardiopulmonary bypass and see how it works. So we started doing in Palmur Lab with initially we did peripheral cannulation and tested the pump, and then now we opened the chest and we did central cannulation and tested it. Sai Kumar will just take you through what we have done in Palmur Lab. Thank you.
Good afternoon, everyone. My name is Sai Kumar. I am an assistant professor in the Department of Mechanical Engineering from Kakatiya Institute of Technology and Science, Warangal. So I was actively involved in this animal studies, which are happening at Palamur Biosciences, Mahabubnagar, in collaboration with AIG. So until now, we have done about 9 studies till now. So 9 animals are done, and initial 3 experiments were introductory animals, and I'll let you know the details of this. So as the pump development is going on, so we wanted to establish the process for animal studies. So for that, before using our developed pump, we have studied how the animals can be cannulated, how it can be managed. So our aim is to manage the sheep for 30 days, the maximum period of 30 days on the pump, on the circulatory loop. It has to survive. It has to do whatever the operations it can do normally, like eating, standing, sleeping. All those activities it has to perform. So for that purpose, we are developing this particular process of the animal studies at PBS. So that is the main aim. So there are various stages initially. So the stage 1, we have performed the sham operation, and as part of stage 2, so arterial and venous cannulation was performed initially at the neck, and as part of stage 3, so left lateral thoracotomy approach, and which was survived for 1 day, and then the period was increased for 7 days, and ultimate aim is 30 days. So this is with respect to the pump. We are also developing the motor as well as console. So next stages are to use our pump, the developed motor as well as the console. So this is the bypass circuit, cardiopulmonary bypass circuit, which is used for the initial 3 experiments. So where the arterial and venous cannulation was performed from the neck, the left side of the neck. So the pump is connected. This is the pump here. Okay, so the purpose of this study is to investigate the effect of the minimally invasive procedure that involves the implantation of the arterial and venous cannulation. So AVR VV ECMO. So the main aim of the process is to provide information of biochemical, biocompatibility related parameters and organ function during this procedure. And this is the circuit. So here you can see this is the pump component, the motor and the pump, and the flow sensor is connected here, and this is the ECMO device also proposed, which will oxygenate the blood, and these are the arterial and venous cannulation. So initially it was developed for cannulation purpose only. So these are the initial cannulation images, first 3 experiments which are performed here. These are the cannulations. This is the pump, the Centrimac pump, which is commercially available, so which is connected to the circuit. And this is the animal which is laying here. So this is the location where the cannulation being happened, and it is also connected to the loop. The blood was circulated. It is a 1-day experiment. And after these studies, we have gone to the next stage, central cannulation So here the left lateral thoracotomy approach was done here. You can see there are inflow cannulas as well as outflow cannula here. So inflow cannula is connected to the left atrium and the outflow cannula is connected to the descending aorta. Here this is the pump and this is the flow sensor, left atrium to the descending aorta. This is the approach which was performed. And during the initial studies, we have faced later a lot of problems, like the animal died even before the cannulation during the first studies. And initially it was survived only for 1 hour. Then later it was increased. So now we are able to perform an operation and we are able to manage an animal for about 5 days successfully. This is our largest survival period. So these are the videos we can. These are the lungs here. Inflow cannulas and outflow cannulas positioning.
Basically, first what we did is we want to, the anatomy of sheep is totally different from human beings. We were surprised, what we found was the external jugular vein was bigger than internal jugular vein. So we cannulated the interjugular vein and passed it to the right atrium. And then initially that is the outflow, and the inflow we put it in the common carotid artery. Just that first two experiments, just to master the technique, see the anatomy of the blood vessels and the heart in the sheep. Second time, and once we were comfortable, we were able to maintain the sheep for a few hours. Or maybe, see, we managed the sheep for 6 hours. After that, we sacrificed it anyhow. No? So then we went for central cannulation. Central cannulation, once again, the same. See, what problems we faced with the sheep was, so we had to keep it fasting. They regurgitate. And one sheep died, when induction, after induction, we were about to open the chest, it died because, it aspirated. So all those things slowly, then we started putting a Ride's tube, trying to take out all the, whatever the gastric contents and all the stuff. So after two peripheral cannulations, what we did central cannulation. We made a thoracotomy, opened the pericardium, put a cannula in the left atrial appendage from which the blood is drawn, and then we put a cannula into the arch and descending thoracic aorta. That was the outflow. And then we brought the cannulas outside and then connected to cardiac gland bypass. Basically, it's basically in the CentriMag, but we used an oxygenator because we are not, initial stages we used oxygenator. That's what we did. And then next is. Yeah. Yeah, so these are the cannulas have been brought out percutaneously through the intercostal space and has been connected to the. So this is the team which has performed the procedure. Then this is the animal during recovery. The last animal was extubated approximately 6 to 8 hours after the procedure, and then we made it stand, and actually we made it. We gave it food also, so it was eating also. So we survived the animal for approximate period of 5 days. So the goal is we want to maintain the animal like this for 30 days. So once you achieve that, then we'll use our Indus pump and test it. That is the idea. So as of now, we have reached this stage, and then we're planning to move forward from July. Once our pumps get ready and all, we'll move forward and further experiment. Basically, sir, what the esophagus? Sir, see, basically, I mean, I'm sure that he will be able to tell.
Polygastric animal, so you
Yeah.
have 4 stomachs, so the passage definitely takes a longer time. I'm not sure, but I did not. You tried the same with the porcines. What was the
Porcine, we didn't,
hassle?
because we want an animal bigger size, like, so porcine, we didn't do it. No, it was not possible. So it chews and keeps the food somewhere inside and regurgitates once again, sir. That's the
Chewing the
problem
guts,
with,
rumination,
that's what they
rumination,
do.
yeah.
They
Rumination.
regurgitate and then chew once again, I eat, I believe, and that's what the veterinarian told us, so. First time it happened, yeah. See, last 3 animals survived. First, I mean, the last 3 animals survived. The last animal, we kept it alive for
No,
5
the
days.
reason I'm asking, it's too precious an animal to lose. 1 out of 4 is 25%. So if there's something we can learn
No,
to
but
avoid this,
yeah,
in
that is in the
future.
learning curve, sir, because
Right,
they are also
it's a
not
learning
aware of
curve.
all. See, the problem is the doctors and at Palmer lab also, they are also not well versed with all this. They usually do all, like some testing of some drugs and those kind of things, but never did this kind of experiments. In whole of South
My
India, I don't
question
think so.
is-
Anywhere they do this kind of experiments, sir. This is the only
Very
place where
nice.
we are able to do it.
Congratulations.
Yeah.
For some reason, these animals are very sensitive, sir. Even a minor changes in any of the physiological changes, they respond very badly. You see, we are involved in another experiment where a leadless pacemaker we are developing, where you are going to plant a device in the auxiliary sternum without anything, and charger as recently we request. Even if you create a minimal pneumothorax, animal is dying. This was first time we had issue on regurgitation. After that, now we are.
What are the flow rates and how much of the cardiac output does it? Oh, that's very good. 3.5 liters. And no hemolysis in this. All the testing was no hemolysis. So when you do the simulation of the flow volume, where is the maximum stasis? You put a 3D simulation and color coded it. Where was the maximum degree of stasis? Where was the hemolysis? On the
So
impact?
usually hemolysis, we did not do the CFD for the hemolysis, and stasis, sir, usually at the outlet of the pump. I can show you.
No, that's where even changing the aortic cannula makes a difference.
So,
Yeah.
here. So usually we observe a ring thrombus here.
Okay.
So we have also 3D printed a device that will come onto the pipe, which will hold it together tightly so that the ring thrombus is avoided.
Like when you take it on upper.
Welding. So the surrogate for hemolysis is where the shear stress is very high. So in 3D simulation, if you say the shear stress is very high, that's where the thing happens. Where is it?
So sir, we fill the loop with the blood so that blood is actually continuously flowing. So we take the blood from the outlet side of the pump, but that would not define the particular location of the pump that is causing hemolysis. So basically, the impeller, the rotating part of it would cause the damage. Yes. This is the device coming on top of the outlet of the tube to tighten and avoid ring thrombus.
Actually,
So my question is, when these animals are being maintained in the lab, what level of veterinary care are they getting? Because this is, a Centrimag is like being in our ICU. Are they getting ICU?
We got a whole team from USA. We got the whole team from USA. What's his name? I don't remember his name.
So, to show the so we conducted a test which the pump ran for 5 days after the surgery and the animal survived. So it was euthanized. Actually, it did not die also. So during that time, for that 5-day experiment, experts from University of Pittsburgh, Dr. Sean Bengston, Dr. Harvey Borowitz, they came and assisted the team at Palamuru Biosciences. So Palamuru Biosciences is basically a very basic facility. It has got all the facilities, but not for cardiac surgery, obviously. So that's why the team from AIG is assisting.
right.
Even the cage in which the animal is kept is particularly designed, and it is, we have got the designs from the US team, and now we are even making it in India. So designing it.
Really, we need to congratulate the whole team for putting together such years of work. All of you, congratulations.
Can you just cut down the live transmission, please?